Hello everyone and welcome back to Mary Notes for Medicos. The topic that we're going to deal today is sterilization and disinfectants. The object is an introduction, classification, physical agents and chemical agents and plasma sterilization. So in sterilization, the process is in which an article, a medium or a surface is eradicated or freed from all the microorganisms in its vegetative or spore states is sterilization. And disinfectants is the destroying of all the pathogenic microorganisms. However, sometimes some disinfectants can't totally eradicate the spores. So these both terms are different. Now the classification is they are divided into three agents or three methods of classification. One is the physical agent, then the chemical agent, and the plasma sterilization. Under physical agents we have heat, filtration and radiation. For heat we have dry heat and moist heat. Under filtration we have membrane filtration, nanofiltration, ultrafiltration, HEPA and ALPA. In radiation we have infrared, UV rays, gamma rays, non-ionizing rays. For chemical agents we have aldehydes, alcohols, dyes, halogens, phenols, gases, surface active agents. And plasma sterilization is a newly invented way of sterilizing. Now we'll be dealing with dry heat. So under dry heat we have flaming. What is flaming? In this process what happens is we use basically materials like inoculating loops, wires, scalpels, spatula, forceps for getting sterilized over here. It is sterilized over a Bunsen flame when it turns until it turns to red hot. Next is the hot air oven. So this is the most widely used method for the sterilization, and it is achieved by conduction. You might have studied about conduction in physics in class 12 and all. So next is the materials that what are the materials that can be sterilized uh, in this. So we have the glassware, test tubes, flasks, silicons, rubber materials, scissors, etc. And all these materials have different holding times, which we'll be discussing in different video. Now we'll be learning about incineration. This is one of the important methods that can help in the eradication or destruction of the medical waste. Uh, but there is this materials known as polystyrene that cannot be incinerated at all. The next is the moist heat sterilization. Under it we are having pasteurization of the milk. In this they destroy all the non-sporing pathogens, and they have two methods basically, Holder method and flash processes. In Holder method what they do is they boil the milk to 60, they heat the milk to 63 degrees Celsius for 30 minutes. But in this the Coxella bruneti bacteria would still survive. But in flash process they heat it for 15 seconds at 75 degrees Celsius, and then rapidly they cool it down to at the temperature of 13 degree Celsius or lower to it. Now this is the vaccine bath. Vaccine bath is basically used to deactivate any sort of bacteria or uh, any uh, any sort of pathogens present in it. For this, the bacterial vaccines are heated uh, at 60 degrees Celsius for 1 hour, and this bacterial vaccines, because of the heat, gets inactivated. And in the coagulable proteins, it can be sterilized at 56 degrees Celsius in a water bath. Now we'll be learning about incipation. Incipation is basically used in Lonstein Jensen medium or serum containing medium for solidification of the egg. This is basically heated for 80 to 85 degrees Celsius for 3 consecutive days. Next we'll be learning about boiling. In boiling, it kills vegetative bacteria if the temperature is maintained from 90 to 100 degrees Celsius for 10 to 30 minutes. Next is the freeze steam at atmospheric pressure. This is performed basically in autoclave. It is used to sterilize any culture media that may decompose if it is subjected to higher temperature. Next is sterilization or intermittent sterilization. So this process is basically used for gelatin or sugars. Here the medium is heated at the temperature of 100 degrees Celsius for 20 minutes for 3 successive days. So what is the principle is, the first exposure it kills the vegetative bacteria, and gradually the spores it germinates after it. So autoclave, what is autoclave? It is a Sterilization by steam under pressure. It is a machine that helps in sterilization by steam under pressure, and it is carried out between 108 to 147 degrees Celsius and aqueous solution at 108 to 126 degrees Celsius. So what are the materials that can be used over for sterilization? Linen, labware, media, and pharmaceutical products. And it is only the biological indicators can monitor the adequacy of the given sterilization process. Next is the filtration. And the filtration, we have membrane filtration. This is made up of cellulose or other polymers. It is used for water purification and analysis, sterilization and sterility testing, and also for the preparation of parenteral use. Here the pore size of the filter is 0.1 to 5 micrometer, and it is most active against bacteria. Pore size is 0.22 micrometer. Next is alpha. Alpha means ultra low penetration of the air. It removes at least 99.9% of the dust, pollen, mold, bacteria, and many airborne particles. The next is ultrafiltration. Ultrafiltration, in this we have pore size range of 0.01 to 0.1 micrometer. Here they reject the particles such as silica, viruses, endotoxin, and proteins. This is a picture of the machine that involves ultrafiltration. Next is the nanofiltration. This is the machine that is involved in nanofiltration. So the pore size is 0.001 to 0.01 micrometer. This is used to filter salt, sugars, dyes, etc. Here, if we see, this is used for filtration of water. Like 3 times, it helps in filtration. 3 times filtered water. Basically, it is used in mineral bottle manufacturers company. HEPA, it is high efficiency particulate air, and it is used to remove at least 99.97% of the airborne particles measuring from 0.3 micrometer in diameter. It is used in operation theaters, laboratories, and biosafety cabinets. Next is radiation. Infrared radiation. This is used for rapid massive sterilization of prepacked items. Example: disposable plastic syringes. UV rays used for disinfecting enclosed areas, etc. Biosafety cabinets. Gamma rays used for commercial plants for sterilizing items like plastic syringes, oils, etc. I'll be going for chemical agents. In alcohols, we have two sorts of alcohols: ethyl alcohol and methyl alcohol. Ethyl alcohol is most frequently used for sterilizing methods. How? Because it acts by denaturing proteins and have no effects on the spore. But it is a better solvent, potent, bactericidal activity, and it is less volatile. When it comes to methyl alcohol, this is effective against fungal spores and is used therefore to clean cabinets, incubators in the hospital, but this vapor is extremely toxic and inflammable. The next is aldehydes. Under aldehydes, we have formaldehyde. This is a markedly bactericidal, sporicidal, and virucidal. This also helps in destruction of the anthrax spores in the hair and wool. Next is glutaraldehyde. This is effective against tubercle bacilli, fungi, and virus. Orthothaldehyde. mm, it is potent bactericidal agent. It cleans endoscopes between the patients, quick and effective and safe. Then we have the parasitic acid. This is a good bactericidal agent. It is active against methicillin resistant staphylococcus bacteria. And next is the hypochlorous acid. This has the bactericidal properties and it is active against biofilms, killing microorganisms within them. Next is dyes. Dyes is divided into aniline and acridine dyes. Aniline dyes is used in Lawson Jensen medium as selective agent, while acridine dyes is used to impair the DNA complexes of the organisms and prevent replication. Next in gases, this is basically used for fumigation. Examples are ethylene oxide, beta-propiolactone, hydrogen peroxide, fogging. Next is the halogens. Two halogens are used, iodine and chlorine. Iodine is used because it is bactericidal and has moderate actions against spores. This is active against mycobacteria and viruses. And chlorine, it is markedly bactericidal and virucidal. But however, Giardia lamblia won't get eliminated if we involve chlorine in it. Metallic salts, we have an example like silver, copper, and mercury salts perform as a excellent disinfectant. Next is phenols. Phenols, it has bactericidal effect due to their ability to damage the cell membrane, and inactive the cell membrane bound oxidizes and dehydrogenase, leading to lysis and death of the microorganisms. The example is phenol carbolic acid that is used in hospital cleanings, etc. Next is the surface active agents. They denature the cell proteins and make the cell lose its permeability. How? By acting on the phosphate group of the cell membrane and entering it. Some of the examples of it is acetyl trimethyl, ammonium bromide, etc. Now, plasma sterilization is a new method of sterilization. It has a radio frequency energy that is applied to a gas, and that helps in the clearing of the electromagnetic field. This is ideal for the sterilization of heat sensitive devices, for example, ureteroscopes, arthroscopes. So that was it for today. I hope you like this video. If you like this video, don't forget to like, share, and subscribe to the channel. We'll be coming with more videos. Till then, stay tuned. Bye.